So, as Aaron said, Teal and I are scientists at the UW. We think a lot about soil and the living things in soil. We're definitely not farmers, so keep that in mind with everything we say. So, just to give you a little overview in case you want to check out, this is what we're going to talk about. So I'm going to spend some time talking about what do we mean by this phrase, soil health, especially from the biological perspective. What does a biologically healthy soil look like? I'm going to hand off to Teal, who's going to give you a little story about how we study soil biology, tell you about some ongoing work in our lab, specifically with the soils from organic grain producers in Wisconsin. And then I'm going to come back and try to end up discuss, maybe as a group, what can we do to improve the biological aspects of our soil. So do we have good evidence for what works, what things are on the horizon, and what should we be thinking about when we're trying something new. So anytime I talk about soil health, I think it's really important for us all to get on the same page, because that's a phrase that can mean different things to different people. So what makes a soil healthy? The big question. The NRCS has their definition of it that focuses on the capacity of soil to function as this living ecosystem to sustain plant, animal, and human life. And so the key concepts there are this continued capacity, right? So soil has to be functioning in the here and now, but a healthy soil will continue functioning that way for some indeterminate length of time. There's another definition that a lot of scientists have used with similar ideas, that it's the capacity of the soil to function, right? Within these ecosystem and land use boundaries, sustain productivity, maintain environmental quality, promote plant and animal health. So there's a lot in common about those definitions, and the one thing that's very true about both of them is how incredibly vague they are, right? So based on that definition, if you handed me some soil and asked me, is this soil healthy? Those definitions give me nowhere to go to answer that question, right? What that really means is that soil health is up to us to define. It's up to you as growers and land managers to specifically define, what does a healthy soil look to you? And the way I would rephrase that is, what do we want our soil to do for us? Right? What are those functions that we want to continue sustaining? All right. This is where we try to get a little interactive. I'm going to hand it off to Teal here to explain how it's going to try to work.
So we want to hear from you a little bit during this presentation, partly because we want to know what you're thinking about already in terms of soil health and what you're doing, partly because you might be interested in what other people in the room are thinking about, and partly because we need to keep you awake for a very long session. So I'm preparing you now, if you can get out your phones. You do not need a smartphone. Anything that can send a text, you do not need to download an app. All you need to be able to do is text. And since we're going to be doing a handful of questions in which you are going to give live information and it's going to populate up here on the screen, just feel free to take some time to get your phone out or go get it in your jacket or whatever, because we're going to be doing this a couple times. I didn't see a lot of movement, but there's some phones coming out.
Be sure we're not recording this data. It's anonymous. We don't know what the answer is coming from what phone.
And the general ideas here. So what you're going to see on the next, you don't have to do this quite yet, but just to prepare you, on the next screen, it's going to, if you have a computer, you can do that first set. But if you have a phone, you're going to, in the to field, just open a new text, and you're going to text in the to field 22333, and then in your message of the text, type OGRAIN. It doesn't have to be capital letters. It can be lowercase letters. As long as it's spelled correctly, you can interact with our screen up here. So let's go ahead and give this a try. We'll do this first one kind of slow as you're getting set up. But we're curious, can you answer this question for us? What do you want your soil to do for you? And maybe one-word answers or short phrases, just a short phrase to kind of give us an idea. Yeah, so people trying it out, that's all right. So we'll see a couple different versions. Sometimes it'll make it into a word cloud or a bar graph. But
that's an excellent answer, right? Like that.
There we go. We'll give a little time to get some responses, and then I'll hand it back to Rick. We have some answers. Fight climate change, retain nutrients.
Gotta have that wow factor in your soil.
And start giving it a try.
I got a second back for a minute. Can I do that? Is it going to screw it up?
I think it'll keep those answers there. So let's try it out. So you're going to get in the to field of a blank text that you don't already have a thread going to your friend. I did that the other day when I was texting. I was texting these random words to one of my friends. I was like, oh, nope, new text, new person.
If it worked correctly, you should get a message, right? You've
Yep,
joined Teal
you've joined.
session, the OGRAIN.
So after this question, just keep your phones out, and we'll do a few more during the rest of the talk.
All right, they're cycling off the screen, so I'll just keep. Did you kick them back? No. Okay. Summarizing it. So I guess I hope we're all seeing some themes. I saw some specific ones about like provide nutrients, retain nutrients. A lot of general ones, right? Keep my family healthy, make money, grow food, right? Essentially, it's all about growing food. All right, I'm going to move on. So you'll have plenty of more opportunities to text and hope everyone's got it working. So what do we want soil to do for us? The way we think about this in our research lab is to think about all these benefits we can get from soil and think about the scale, both the spatial scale and the temporal scale of those benefits. So some things we want soil to do work well at the very local level, like in your field, right? You want to do better at holding water, maybe you want it to provide more fertility for your plants so you can reduce your fertility inputs, reduce disease pressure, suppressing diseases. Those are local scale benefits. But healthy soil could also have broader scale benefits for all of us outside of your farm. So reducing soil erosion, reducing nutrient and pesticide runoff. Someone said fight climate change on I saw that scrolling through. So things like store carbon that helps modulate the global carbon cycle. So healthy soil, what you do on one farm could have big consequences for larger scales. We also have different benefits at different times. So you can think about benefits from soil that occur in that one growing season. So you reduce disease, or you reduce fertility rates. You can think about slightly longer term benefits, where you're thinking about maybe a whole rotation scheme, a 4 or 5 year cycle, where you want your soil to be promoting good outcomes across that whole rotation cycle. And then really long-term benefits, right? If you are thinking about who am I going to pass this land on to when I'm done farming it? Is that going to go on to someone else in your family? You want to maintain healthy soil that continues to be healthy for future generations. So what makes the soil healthy? As I'm sure you guys are aware, soils are very complex systems that have physical, chemical, and biological properties. And all these things work together. So it's actually very difficult to pull out the physical properties of soil. That affects the chemical properties, that affects the biological properties, and vice versa. So because soil health is such a vague concept, and because soil in general is so complex, we don't have a one-size-fits-all measure for healthy soil, right? Instead, we measure a bunch of indicators. So there are some indicators we can measure that tell us something about the physical properties of soil, and there are Many of these are fairly well understood. We know which direction we want them to go in. If we have more aggregate stability, we're likely to have better water holding capacity. That's generally going to be better for plants and crops. These are nice because you can send them off to a lab to test to get quantitative measures of them. You could also look and see and feel these in your soil. You can get some sense of, is my soil really loose and falling apart, or do they have these nice clumps that stick together? And we know a lot about the chemical properties of soil. Again, you could easily send these off to a lab and get precise numbers on things like the pH or the levels of micro and macronutrients in your soils. And not only can we measure them accurately, we have some good understanding of how these levels correlate to crop yields and plant health. Right? We know what we want more of, what we want less of, if there are optimal ranges for many crops. Key thing about these physical and chemical properties is that the levels that you'll have in your soil reflect both inherent properties of the soil. So that was mentioned in the previous talk a little bit, like same bedrock material going to affect what chemical levels or what the physical structure of the soil. If you're on a sandy soil, there's not a lot you're going to do to change that. But they also affect management decisions. So how you're managing fertility, your tillage practices, all that will change these physical and chemical properties. What Teal and I are going to talk most about are the biological properties of soil. So again, we have these indicators that you can get measured in labs, like the organic matter in your soil, the mineralizable nitrogen in your soil. That's a lab assay. Some labs will measure things about soil enzymes, which are proteins produced by microbes that break down chemical compounds in soil to make other nutrients available to plants. You can get these gross measures like soil respiration, which is just a measure of basically your soil breathing. All the microbes in there, how much carbon dioxide are they exhaling in an hour or a day or whatever. You can look for certain organisms like earthworms, those are easy to find. So when we come talking about the biological aspects of soil and the indicators, key thing to think about here is that these are really poorly understood. Especially how they relate to crop health and crop yield is poorly understood. Microbial soil respiration, for one, that's one many people measure. We don't know. There's no consistent relationship. Is more respiration mean good crops or bad crops? Actually, that relationship is not clear, and it changes based on the context. All right, so all these biological indicators are a gross oversimplification of the biological communities you find in soil. So there's just some factoids from NCRS about soil biology. You pick up a handful of soil, you have hundreds of millions, billions of individual organisms in that, microbial cells. And tens of thousands of distinct kinds of microbes. So snapshots like organic matter or potentially mineralizable nitrogen are not going to capture that kind of diversity. This is where folks like Teal and I are coming. This is what we're trying to understand is what about that complex diversity is really important? What do we want more of? What do we want less of? Can we develop tests or indicators that do a better job of capturing all that stuff? But it's actually really difficult. So I'm sure you're all aware, soil microbes can affect crops directly. And so this is my little cartoon of a plant to give just a sense of the different kind of groups of microbes that might be interacting with that plant's roots. So you've got things like saprotrophs that are just living freely, degrading the organic material in your soil. And then you've got other microbes that are much more intimately associated with the plant. So if you're growing a legume, there might be rhizobial bacteria forming nodules on those roots. Most crop plants can have the least potential to form interactions with symbiotic fungi called mycorrhizae. And then, of course, all plants are susceptible to a greater or lesser extent to microbial pathogens. Right? So they can affect crops directly, but they can also affect crops indirectly by changing how plants respond to abiotic stresses. So the microbes you have in your soil can have an effect on how drought tolerant is your plant going to be, how will it handle low nutrient stress. And so when we think about biologically healthy soils, it's not just about the microbes that are directly interacting with your plant, it's also about the microbes that are mediating how your crop responds to all these abiotic stresses. So clear examples there for nutrient acquisition, right? So we've got microbes like mycorrhizal fungi that basically act as extended root systems for the plant. So they're growing out of the plant's roots like these little straws sucking up phosphorus and nitrogen from the soil, and in many ways are more efficient at doing that than a plant root would be. If you're growing legumes, they might be associating with rhizobial bacteria, right? And those rhizobial bacteria are actually getting them direct access to the nitrogen present in our atmosphere that most other plants can't access directly. You think about drought resistance. I talked briefly about soil aggregation and water holding capacity. That's not only a function of the chemical and physical properties of the soil, that's also a function of the biological properties of the soil, because fungi in particular bind soil particles together. So the higher, more fungi, more fungal hyphae you have growing through your soil, they'll act like a net to hold soil particles into these clumps. Some fungi even exude these like protein glues that like glue soil particles together and make those aggregates more stable. That's sort of an indirect effect. Again, there are also direct effects. So those mycorrhizal fungi growing out of your plant's roots can directly access little pockets of water that a plant root can't get on its own. And then, I'm in a pathology department, so I think a lot about disease. The native microbes in your soil have a lot to do with how badly, how much disease your plant gets, even if the pathogen is there. So soil microbes can inhibit soil-borne pathogens in sort of general ways. Just having a lot of microbes out there can compete with the pathogen, or you can have really specific ways where you have a specific microbe in your soil that inhibits some particular pathogen. So the best example of that is wheat take-all disease, where if you grow a monoculture of wheat over and over and over again, your levels of disease go through the roof, but then at some point you break through it, and that take-all disease essentially goes away. That's something we observed over and over again, and now we understand why. It's because there's a particular group of microbes that develops over time in that wheat monoculture that inhibits the take-all pathogen. So we've got all these different microbes, these tens of thousands of species of microbes in a gram of soil. They all have these different effects on plant growth. They're not all good, right? So clearly there are microbes. Pointer? No. There are clearly microbes that reduce plant growth, right? We all know about the pathogens. There are microbes that do a great job promoting plant growth. And then there are a lot of microbes somewhere in the middle. But if you take nothing else from this talk, I want you to consider this, is that one of the reasons it's hard to study soil biology, one of the reasons we don't have clear, straightforward recommendations for growers is because everything is context dependent. So take your textbook example of a beneficial microbe, a mycorrhizal fungi. You would think everyone wants more of those. So the way that fungus works, it grows into the plant's roots. The plant feeds the fungus. It gives it sugar. In return, the fungus grows into the soil, collects phosphorus and nitrogen for the plant. That's a really beneficial relationship for the plant if that plant needs nitrogen and phosphorus. If that plant doesn't need it, if you've given it all the nitrogen and phosphorus that it needs, say you've put chemical forms of it down, that can just go straight into the root, right? That fungus has nothing to offer the plant. It's still going to want the carbon from the plant. The fungus still needs to live. So it's pulling sugars out of the plant. It's not giving anything of value in return. So there are certainly situations where having mycorrhizal fungi reduces plant growth. It's all about the context. All of these relationships are a give and take between the plant and the microbe. If what the microbe has to give, the plant doesn't need, then the plant's better off not having that microbe. So when you're thinking about what what biology do I want in my soil? What microbial community do I want to have in my farm? The answer to that question depends on everything else about your soil and your farm. What's the nutrient status in that soil? If the nutrients are very readily available, then you don't want these nutrient-acquiring microbes. They're just going to be a drain on your plant. But if the nutrients are bound up, if you've got organic kinds of nutrients, then you need microbes to process those before the plant can use them. In that case, you really want these guys. So yeah, so even beneficial microbes, in quotes, can be detrimental in the wrong context. So always keep that in mind. So there's still a lot we don't know about soil biology, and beware of anyone who tries to claim otherwise. Like I say, I hear a lot on the industry side from researchers making strong statements, and it always makes me a little uncomfortable as a scientist because there's still a lot we really don't know. And all the answers are context dependent, which makes it really hard for one person to say, this is the answer for you. So here are some of the key questions I think we still don't have clear answers for. You know, we have the abilities now to measure those tens of thousands of microbial species. For a long time, that was sort of a black box. We knew they were down there, but we really didn't have any tool to identify them. We can do that now. We have DNA-based methods that can tell us which microbes you have in your soil. But where we still don't, what we still don't understand is how that composition of the community, which microbes you have, relates to the function of that community, which is what we want to know. Is it making nitrogen, speeding up the rate of nitrogen mineralization? Is it suppressing disease? Is it generally making healthier crops? That relationship still needs a lot of work to untangle. We're still just beginning to understand how microbial communities respond to farm management, both in terms of their composition, which microbes do you have there, how many different kinds of microbes do you have, and their function, because we still don't quite understand how composition relates to function. So if you change your tillage practices, how's that going to affect how the microbial community in your soil functions? Is it in a good way or a bad way? And then following from that question, what should we do? If we decide this is the kind of microbial community we want for this kind of cropping system in this soil type, in this state, what do we do to move from where we are to where we want to be? So I'm going to hand it off to Teal now, just to give you a little glimpse of how, in my research lab at UW, we're trying to answer some of these questions specifically for organic grain producers in southwestern Wisconsin.
Thanks. So I'm going to talk to you for about 10 minutes about some research I'm leading in the lab, a set of different experiments, and I just started 6 months ago, so I am excited. We do have some results to share with you, but I think you'll also be interested in the general approach we're taking, and we'll have a lot more in the next year as we're getting through the data. So the largest question in the study that I'm leading is generally, how do organic practices change soil over time? So a really broad general question, and we're looking at whole microbial communities in the soil in our lab as opposed to individual pathogens, which is more typical of a plant pathology group. And this is a hard question to answer if you haven't been monitoring the same fields for 30 years. I haven't been doing that, obviously. So we're taking a different approach so that we can ask a question about how is soil changing over time to understand how that might be changing these attributes that we're interested in, water retention. I'll throw an example up here. So we're using soil from a bunch of different farms that farmers have donated, growers have donated to us, to get at this question. So on this bottom axis here, we've organized, these are soils, basically, soil samples from recently transitioned. We have some transitioning 2017, back longer than 20 years ago, and we'll be getting some more this summer. And with this, we can start looking at how do these properties differ among these farms that have relatively similar soils from the same region. And we don't know this, right? organic management of soils hasn't been for long at all when you're considering how long soils develop over thousands of years. So there's a lot that we can just learn using this basic approach. Do we still see accumulation after 20 years of some of these things we're interested in? Or do we see sort of a plateauing? Is it, we're ramping up after 10 years and then you're good to go? Or maybe something else is going on for some of these attributes where, hey, we see this ideal peak around this time period, and then we've got to maybe think about shifting our management because it seems like we're decreasing. We don't know these questions. Maybe some of you have predictions based on your own experience. Maybe some of you are just transitioning and thinking, oh, this could be interesting for me. So this is one approach that we're taking. We're doing a field experiment, as I said, and instead of just a few farms, we're collecting soils from over 90 fields, dozens of farms, so that there's going to be a lot of noise here, right? There's going to be a lot of variation, but hopefully with this many we can start to see patterns and actually have some answers that we can generalize, because we're not just doing a study on one single farm that may not be useful to somewhere else. And we're collecting a lot of data. We're analyzing physical, chemical, and the biological attributes. So in addition to soil organic matter, one of the things I will be doing is sequencing, getting the DNA code from all of the microbes in all of these samples and seeing how does the microbial community differ. And then we're doing this other experiment that I think you'll be interested in, to actually see how those microbes impact things like plant yield. So that's where I'm going to next. I don't have data yet to show from this part, but this is sort of looking how the soils, different soils compare to each other. And what we're really interested in as a lab group is focusing in on the microbes, mainly bacteria and fungi in the soil as groups. And we want to know how these microbes are actually impacting yield. And it's complicated because the microbes are responding to the management, right? All of these things are changing in the soil all the time, but they're also influencing things like pH and potassium and nitrogen availability, right? So we have developed an approach to isolate the effects of the microbes that is not at the same time looking at the effects of pH and organic matter and all these other things. So that's what I'm going to describe now. Soil management affecting those things too at the same time. And you have this changing, the microbial community is changing all the time. So in this approach, we can actually take those soils from the different fields we're collecting and use the microbes that are different from all these fields to see how they influence yield in a greenhouse experiment. And part of this approach that we're using involves this crazy machine that I wanted to show you. This is me opening up a giant autoclave in which we pressure cook the soil that we're using for this experiment. So, I know it sounds weird, but most of the soil in all of the pots that we're using is going to be a mix of sand and local soil that is actually pressure cooked, so we're killing most of the microbes in it. So most of the soil in all the pots is going to be the same, and then we're adding just 5% of the soil in the pots will come from these different fields. So we can actually add these different microbial communities, but the plants will be experiencing really similar soil texture, similar pH, and things like that. They're going to all have different microbes that they're interacting with. And so to add a little more complexity, we want to know why, what are the microbes in these different fields doing differently? What are they good at if they come from farms that have been under organic management for longer? So another aspect of this experiment is we're adding different types of organic nitrogen and urea as a comparison of varying complexity from left to right. So left, the corn or plants that we're using, the crops that we're using have access to that right away, right? You don't need microbes. But in organic farming, you need the microbes to process and break down these different forms of nitrogen. And you need different microbes to process these different chemical compounds in these materials. And it's also interesting when you're working with these, you're not just adding nitrogen, you're adding the whole suite of nutrients that comes with those different manure. We're adding red clover and sorghum Sudan grass in this experiment. So we might think that in an organic system, microbes from farms that have been under organic management for longer might do a better job at decomposing these more difficult, complex materials like sorghum Sudan grass or rye from your cover crops even. And so there's a lot more steps though, and we want to see, how do these differ? And additionally, how are the microbes providing nitrogen differently to different crops that you might have in rotation? So in addition to this set of nitrogen treatments, we grew this fall rye, soybeans, and corn, all organic varieties. And so this is our experimental design. We have every combination of every crop species, every nitrogen source, and multiple replicates, so multiple plants grown in each of these. And we have 10, I believe, different field soils and then 2 conventional for comparison. And so here's just what it looks like. At one point when I was developing the experiment, so adding the clover, the sorghum Sudan grass, and then manure, and then we added the soil on top of that, microbial communities. Here's a photo of the experiment as it's growing. It ended up being 550 pots. It was a big experiment for me. And we harvested everything but the soybeans. They're still going, but we recently harvested some, so I do have some data to show you. Just a couple quick results, and we have a lot more coming. So one thing that we saw, a pattern that we saw, that the farms that have been managed under organic for longer had higher corn height when they were grown with the sorghum Sudan grass. So that's kind of interesting. This is, like a V4 stage, so because we couldn't grow the corn all the way to maturity. So we're just looking for differences in growth for this particular data point. But this is interesting. That means that organically managed soils perhaps have microbial communities that are good at accessing that really complex nitrogen source. So that's interesting, and we did not see any of those patterns with any of the other types of nitrogen. So a lot more to dig into here, but we thought that was interesting. And one other thing we noticed as we grouped the data by whether the fields where we collected the soils from had a rye cover crop last summer or not. And we grouped them by that, and we found that regardless of what nitrogen we added, and those are the colors on the side, their corn height is taller in the soils that were given microbes that had a rye cover crop. And so that's kind of interesting, right? Because you're not adding the rye cover crops to promote microbes that are going to help your plants grow necessarily. They're doing it for other reasons, but there might be this added benefit too. So these are not, this is corn height is all I'm showing you right here because that's the data that I have right now. But we have a lot more coming. And I guess lastly, just to remind you, this is interesting because, we're not adding the rye to these pots. We're not adding nothing about that soil besides the microbial communities should really be affecting the differences in growth here for the corn, right? It's the actual microbes that were associated with those rye cover crops, given the experimental design. And so just to show you again, here are all the combinations that we are getting the data back from and analyzing. We're measuring growth, chlorophyll content, and then yield for soybeans, so we can have some different things to look at that will be more interesting than corn height. And here's the data collection stage. We have to still weigh all of those paper bags full of dry plants and sequence the DNA on all those tiny little roots to get the rest of those data. And so With that, I'm going to hand it back to Rick, but this is the general approach that we're taking in the lab and doing with a couple other experiments as well. Thank you.
So hopefully maybe we can come back here next year and give you a more complete story of what we learned from that bigger project. And we didn't point out that this project is part of a larger group. Aaron's the leader of this group, actually, as well as Matt Ruark in the soil science department, and lots of other people. So Teal and I are just doing one aspect of this larger project, trying to understand how soil health in the broadest sense is changing over time under organic management. So I have a little bit of time left. And I wanted to say what I can say and what I can't say about, practices. What can we actually do to affect these changes in soil communities? And so I hope many of us would agree that this is where we want to go, right? So if you think about many fields now, maybe not all of them, imagine that they might have a relatively low microbial function because of the history of inputs, of disturbance, of everything we do in intensive agriculture. And many growers, sort of compensate for that low microbial function with higher inputs, right? So we have chemical inputs to make up the differences. And you can do that and you can get high yields. But wouldn't it be better if we could have the biology of our soil taking some of that off our hands? Right? So can we get to a situation where you have higher microbial function, and so you would need fewer inputs, and you can maintain those high yields and increase your profits because you're not paying for the inputs. So how can we get there? Can we increase the soil microbial function through either direct or indirect manipulations? So when I say indirect manipulations, that's just what we're doing, right? Our farm management. Everything you do on your farm affects the microbes in your soil, good or bad. That's sort of inevitable. Direct manipulations would be trying to actually put a microbe in there. So in terms of indirectly affecting soil biology, the recommendations for that are just these general good agricultural practices, right? That, especially those of you doing things organically, are certainly already doing many of these. So this isn't coming from me. This is just in RCS. It's to some degree sort of common sense. If you can reduce disturbance, you're disturbing the microbial community less, and so it'll have more time to develop that complexity. If you can add biological diversity, so diversity of crops, diversity of other organisms that live on the farm, insects and all that, through time in the rotation, so having more diverse rotations, or having mixed plantings, doing a cover crop cocktail of some kind where you're planting multiple species at the same time. All of that is adding biological diversity. And one of the general rules of thumb in ecology is that diversity begets diversity. If you add more plant diversity, you're very likely to get more microbial diversity, and vice versa. Doesn't always work out that way, but as a general rule of thumb, that's pretty safe to say. The other key recommendation is that keep a living root in your soil as much as you can. So again, soils are complex. If you drill down to what's happening at the scale that a microbe would pay attention to, soils are not homogeneous. Think about even in like a square inch of soil, there are deserts and oases for a microbe. There are places around a soil particle that's essentially a microbial desert. There's not a lot of food there. It can get wild extremes of moisture where it gets flooded and they can't breathe, and then it dries out and they get desiccated. The area around a root that we call a rhizosphere is that oasis. High nutrients for the microbe because the plant is like exuding sugars and things that microbes live off of. It's buffering that environment so the moisture content doesn't get too high or too low. The oxygen levels don't get too high or too low. The pH is buffered. So that's where a microbe wants to live. And if you look at the density of microbial cells or microbial activity around a root, it's orders of magnitude higher than it is even, a micron away from that root. So what that means is if you have a live root in your soil, that's making microbes happy. If you don't have any live roots in your soils, the microbes are sadder, right? Many of them are going dormant, certainly not growing. So the longer you can keep alive living roots in your soil, you're giving that comfy environment for a microbe to live in. And then the other general recommendation, right, is that you want to keep your soil in place. It's hard as a microbe to live in soil if your soil is blowing away in the wind, so reducing erosion has many good properties, but one of them is that it's keeping your microbes happier. Okay, but I said everything's complicated when you talk about soils, and that's true even for these very general recommendations. Because soils are so complex, management practices can have unintended consequences. So we know tillage disturbs soils, and I say, oh, that's bad. But it also can act to incorporate that organic matter that's on the surface of soil deeper into the soil so that microbes have access to it. It can take soil-borne pathogens that are deep in the soil and expose them to the air. Well, they will dry out and die. So nothing's all good or all bad when it comes to a complex system like soil. And cover crops, which I'm a huge proponent of, and I was just telling you how important it is to keep a live root in your soil. That's certainly true. But we know that there are potential downsides, right? There can be yield drags with cover crops because they're depleting, those cover crop is taking up nutrients from the soil. And if those nutrients aren't getting effectively recycled back into the soil, then your cash crop might have less nutrients to begin with. So unfortunately, it's hard to make conclusive statements. When you're dealing with a complex system, always be aware of the intended consequences and the unintended consequences. So with that in mind, I'll give you guys a chance to inform us a little bit. So it's easy for me to do this research and say, you should do X, Y, Z. It would be really helpful for us to know, what are your biggest challenges that are preventing you from following some of these general recommendations? And that would help us know what should we try to tackle as researchers. So I'll give you guys a few minutes if you want to. This will be like the other one where you can write longer things and they'll just start scrolling. And we'll have time for more questions at the end, so I won't take too long on this. I'll say that particular research project where we're looking at time under organic management, just be aware that that variable means a lot of things, right? So obviously it's not years. Years are just numbers. It's what's changing over that time. And we will do our best, but I doubt we'll be able to drill down and say, this was a specific change that was important, because it's probably lots of things. So it could be about chemical residues and how long it takes them to go away. It could be about other general changes in organic matter as it builds up or brings down. And we, at this point, can't say anything about that. Okay, have you been keeping? Weeds, I know that was going to come up there. That's a big one, right? Oh, that makes me feel good, knowing what is best to do. That's, as a researcher, we're like, hey, we have a role to play. If we can give you an answer, don't come up here and say, we don't know either. Testing, that's another thing that my lab is working on as well, is can we develop informative tests about soil biology? So we don't know what to do, but at least we'd be able to track whether what we're doing is changing things in a good or a bad way. Aaron can talk about things like cover crop termination, right? All right, I'll take one more question then I get. Yeah, and we know a lot more about the macronutrients, nitrogen, phosphorus, potassium, than we do about the role of micronutrients. And there are some strong proponents of certain micronutrients as being understudied. And that's another thing that's going to be really context dependent. What's the right level of calcium for a soil? There's not an answer for that because different soils are going to have a different optimal level, right? Or what's the right ratio of calcium to magnesium? That's something that's going to have to be determined regionally or for a given soil type. Okay. I'm going to cut this off. I hope that was helpful. All right. So I don't know if this will be of interest to you or not. We only have a few minutes left, so I won't dwell on this. But I do get asked this a lot. When people find out that I study soil microbes, one of the first things I ask is, should I buy this thing? Somebody was trying to sell me a bag of something, a bag of microbe X. Should I buy it and put it in my field? So I've thought a little bit about that, and I don't have any answers, but I'd give you some thoughts about what my thoughts on that are. So it's come up because this is a growing business, right? So that's a quote there from Hugh Grant, who at least at one point was, I don't know if he still is, chairman of Monsanto. So if Monsanto's getting into something, then some people are really thinking about it, right? They think, at least they think there's some money to be made here. And interestingly, one of the economic pushes for the big biotech companies, agri-tech companies, to get into this microbial products industry is that it's much less regulated than the GMO industry. So for them to bring a new GMO onto the market is a really long, expensive process for a big company. To bring a new microbial product to the market is much faster and cheaper. So they're thinking, maybe we can do this and make just as much money as we have off our GMOs, and it'll be much faster. So they're now putting serious efforts behind it. And the industry is really growing, right? So those are just some numbers. Don't pay attention to the numbers, just the bars are getting bigger, right? Some of those are actual data, some of those are projections of how much do we think the world is going to be spending on these microbial products. So I'm interested in this group. You guys can chime in. If anyone is actively using what I'll call a microbial product. So that's when you buy something that's intended to be a living microbe that you would add to your soil. Any kind of living microbe. Yeah. I'm not keeping track anymore of how many responses we have. Okay, I'll wait until those numbers, they seem like they're stabilizing. Because everyone who grows soybean is doing. Okay.
You can change.
Can I reset it?
People can change their answers. They only get one answer recorded.
Oh, okay. So if you, all right, I'll take a look at that number. All right, that's polluted with the legume seed treatments. Now go back, and if that's the only microbial inoculant you're using, change your answer from yes to no, and it should record that. So if you're using any microbial product in addition to your rhizobial inoculation for your beans, say yes. And if that's the only thing you're using, say no. So see how those numbers change. Okay. All right. We have a feel like that's a good sense of the room. All right, so that's enough that I don't feel ridiculous with my going on to the next slides. So good. If it had been like 100% no, I would just stop now. So people are doing this. All right, another question for you. So now think about, again, let's cut out the rhizobia, because it seems like everyone's doing that, and we know why. For all those other things you're adding, what are you trying to get with that microbial product? What is your intended goal? And this is just you can type in words. So you might put disease suppression, maybe, or biocontrol or something like that. Or you could put nutrients, biofertilizer, general answers like healthy plants. Seems like that's what we all want. Yeah, yield. Of course, we all want yield. Get as specific as you can. That would help us, right? Okay. Biocontrol. So we've got some nutrient-related answers and some pest control-related answers. And then a lot of answers that are in this vaguer concept of, like, we want things to be better, right? We want healthier roots. We want greater biology, higher yield. Well, I did ask you to be specific, so I assume everybody wants to be specific. Okay. And then we had this. Now, you'll have to answer again, so I apologize for that. My hope was to try to categorize this a little bit. So think about your answer you just put in there, and try to put it in one of these camps. And if you can't put it in a camp, you can put it down there in that D. So you just have to type the letter, right? Okay. Is that everyone? Is that 2 people? Like, it can't be that perfect. All right, people are getting, what? Oh, no. Sorry. Okay. Well, we'll move on. Based on the answers there, I feel like a number of those answers, a few of the answers I would have put in that biocontrol camp, so if anything you're trying to do that's related to pest control, reducing disease. A few I put in that biofertilizer camp, which would be anything you're doing nutrient related, whether it's, some microbes are intended to help your plant access nutrients, so like a mycorrhizal fungus. Again, it's just like a straw connected to your root that's like sucking up mineral nutrients that are in the soil solution. Other microbes are actually doing the work of decomposition where they're chomping up organic material to release nutrients. I put growth promoter up there in quotes because that's what I see a lot marketed, right? So on the bag, it'll say this is a plant growth promoting rhizobacteria or something like that. And one of my goals with this part of the talk and in my research is to try to break that down a little bit. So I'm going to cut you off here. Excuse me to anyone still working. A little bit what they mean. So microbial pesticides, or those biocontrol agents, that should be pretty clear. This is a live organism that's intended to inhibit another live organism. So there are quite a few of these on the market. Some of them will make broader or more narrow claims, like this will suppress this specific disease on this crop. Others will claim to suppress a lot of different diseases. And we got this broader category of microbial soil treatments. Some of those which I would consider biofertilizers. So they're seeking to increase nutrient availability to your plant. I would put mycorrhizal fungi or rhizobial bacteria in that camp. There are some products selling what are actually free-living rhizobial bacteria. So these are bacteria that can fix nitrogen from the atmosphere, right? We know much about how that works for legumes, where they actually host them in their roots. They give them this comfy little nodule to live in. But there are bacteria with that function that live freely in soil and can be fixing nitrogen from the atmosphere, and just it's there in the soil. That's generally a relatively low-level process in most soils. Some companies are trying to sell microbes that do that with the intention that they would actually be doing that in your soil. It's pretty questionable whether they do. And so the question I get asked a lot is, will this work? If I buy this product, will it work in my soil? And of course, I don't have an answer for that. I hope that's clear from all the other slides I've given, that we don't have answers for anything when it comes to a particular soil in a particular field. But what I do have is a list of questions you could ask that help you make that decision a little bit more finely. So if it's going to work, two things have to be true. It's got to successfully integrate into your microbial community that you already have in order to perform its function. And it's got to achieve some additional thing that your microbes aren't already doing for you. And you always have to keep in mind this context dependency. So just because something worked in your neighbor's field is no guarantee that it's going to work in your field, right? Because it's going to be going into a different context. And in fact, something that was beneficial in one field could actually be detrimental in a different field if the context is different. So here's what I think when someone asks me this question. Well, what I want to know, does my crop need this organism? If the answer is no, then I should stop there. Is this organism already in my soil? If the answer is no, you should stop there. If that organism's not there, is there some other organism that's already doing that job? Again, if the answer is no, you can stop. Then, if you get down to those first three questions, you can say, well, if I add it, is it going to even survive in my soil? So there's no point in just throwing a microbe on there that's going to immediately die. And then, even if it can survive, can it compete? So your soil is already full of microbes. So if you're trying to add a new microbe in there, it's got to find a place to live. It's got to fill what we call a niche in ecology. It's got to be able to do something that allows it to persist, even though there are other microbes trying to use those same resources, right? So it's got to be able to compete. Or otherwise, it'll just get kicked out. And so as you undoubtedly are thinking, as you think of these questions, like how in the world would I answer that for my soil, right? Many of these are not answerable questions. But some of them are. So does my crop need this organism? That is something you should be able to answer. And the person trying to sell you the thing should be able to guide you to that answer. Because if they're selling you something, they should be able to tell you, what is this going to do for my crop, right? And this is where I get a little suspicious of the generic plant growth promoting microbes, right? How is it promoting growth? In what specific way is this going to make your plant healthier? You need to know that to be able to ascertain, does my crop need it? If it's about nitrogen, what's the nitrogen status of your soil? If your plants are already, is there any evidence that your plants are deficient in nitrogen? If they're not, then a nitrogen acquiring microbe is probably not going to do you a lot of good. Same about the biocontrol agents, right? Do I have a problem with this disease? Are there other ways to manage that disease? A good company should have answers to these questions. So that first one, maybe you guys can answer. And then also always be aware of basic crop microbe incompatibilities, right? So it sounds like this is an educated enough audience that you wouldn't try to put our isobial product on corn. You would know that's sort of pointless, right? Corn can't make nodules. Sometimes, this may be not super relevant for this group, there are plants that won't form connections with mycorrhizal fungi. They just lack the genetic capability to do it. So anything in the cole crop group, broccoli, cabbage, anything of that, canola, if you're growing that, or mustards as part of your rotation or the cover, those are not hosts for mycorrhizal fungi. So no point adding your microapply product to that crop. Not going to do any good.
There is a car on the loading dock that is blocking a Pepsi truck from entering. It's in a plunder, and the license plate is ACB2364. If it's one of you guys, you should move the car out of it. Yeah, it's going to be towed otherwise. It's a plunder, ACB2364.
Anybody running out the back? Okay. I'll get quickly here. So the rest of these questions are harder to answer. So is the organism already in my soil? We don't know which organisms are in our soil. Is there another organism doing that job? Again, we don't know that. There is new technology coming on board that probably in not too many years you could get a soil microbiology test that could tell you a list of what microbes you have in your soil. So you could at least check it off, like, oh, I already have a lot of it. These are the hardest questions to answer, because even if you don't have that microbe in your soil and you think I should try adding it, there might be a reason why you don't have that microbe in your soil, right? Because it can't survive there. So probably unanswerable, but you should at least ask the seller, the producer, if they've done their homework of testing this product in a lot of different kinds of soil, showing that it produces positive effects across different soil backgrounds, especially soils kind of like yours, right? So have they been tested in anything like your soil type? If not, be suspicious. But ultimately, the only way you're going to know is if you test it in your own field. And so ultimately, that ends up being my answer to anyone who asks this question as well. Buy a little bit of it, try it in a corner of your field, see if things look better in that corner than the other ones. Best way to do that is to do it not just one time in one corner, but do it in a couple places, maybe do it in a couple years, see if that effect is consistent. So what can we do to improve soil? The biological aspects of soil health on a farm. Unfortunately, we don't yet have definitive answers, but there are some general suggestions. So again, that rule of thumb, diversity begets diversity. Anything you can do to maximize the biological diversity on your farm is likely to lead to higher microbial diversity, and higher microbial diversity is likely to lead to higher function or more stable functions across the board. I don't want to sound completely sort of nihilistic about microbial inoculant products. I think they potentially have a role, but consider them for specific purposes and specific needs. And evaluate whether those specific purposes are needs that your farm has. And just beware that anything we talk about with soil biology is unpredictable. And it depends on the specific context of the soil. Because it's so complicated, there are a lot of ways it can go. So always just enter into that with eyes wide open. Right? All right. And I will stop there, and I think we have a little bit of time. Happy to entertain questions if anyone has them.
I take a couple questions before we move on to the next talk. So if you can repeat the question, Rick.
Sure. I was just asking this specifically in our research projects, you mean what in the soil food web are we focused on? And our particular expertise is in the microbes, so the bacteria and the fungi, partly because those are the least understood. That's not to say that they're the only important ones, right? And so in the larger soil health project, I think we will make some effort to understand, what's happening with nematode populations, earthworms, soil insects and soil fauna. All of that's important, and we should at least keep an eye on those, right?
On the dry soil microbes, when you inoculate, is there carryover? I mean, can you get one inoculation and then you'll have a soybean microbe?
Right. So
repeat that question again.
Yeah, so he asked me specifically about legume or soybean inoculations. You're inoculating rhizobia. Do you have to do that every time you plant soybean, or by doing it once, do you build up a population that's still there when you plant your next round of soybean? That's a really good question, and I shouldn't speak too off the cuff here, but I know even in Wisconsin we've had some other research labs who've tested soil for the rhizobial population there, and they've always been high. So you should consider, do I need to buy that rhizobial inoculation? You may already have a high, healthy rhizobial population in your soil. We'll pick that up in our little greenhouse experiment, because we've got soybean growing, and we did not inoculate them with rhizobia. So anything that any nodules they make, it's because they found that bacteria in the soil we gave them, which is coming from farms. And we haven't looked at the roots yet, but the plants look pretty healthy and pretty green. So I'm pretty confident they've got nodules.
Asking on what is the coldest day of winter, but basically, what are seasonal effects? I mean, you have winter, you have a frost layer, and I would think this is perhaps a large effect on the soil microbial. But what are you finding? Is this a variable you're looking at?
Well,
Repeat that question.
so he's asking, how do the microbes change over the year? How do they respond to seasons? And maybe another way to phrase the question is like, all microbes in Wisconsin, the microbes you have in your soil evolved in Wisconsin, so they can deal with winter, right? Otherwise, they wouldn't be here. They have strategies to go dormant when soil's really frozen. But that doesn't mean that what happens in winter is irrelevant to what happens in summer, right? So maybe they can deal with winter in general, but can they deal with a really cold stretch, an unusually cold stretch? What does that do to the microbe? Does it kill some off? Or maybe more concerning, what if they don't get the kind of cold winter that they have had in their millions of years of evolution, right? So if the level of snowpack changes and we have more exposed soil because we have less snow, that actually makes the soil colder. I don't have a great answer for that yet. I say that a lot. But it's something to keep in mind that in general, microbes, like other native organisms, have had to deal with variable weather in the past. And so they have evolved strategies to do that. But if things are changing outside of their sort of the bounds under which they evolved, we need to be considering whether that's pushing them outside of their tolerances.
I think that's a great point, and I'll make the last point before we go into, but I think, and I too, like Rick, I mean, I don't want to be totally negative about these applied products. So obviously, rhizobium bacteria have a huge benefit in terms of legumes, and there's some other products that really have pretty consistent results. But I do think, more and more we know about how there's regional adaptation to crop genetics and looking at fostering native species. So as we think through this, an application of inocula that we're looking at might have the same effects, not only across soil types within different counties, but across the country. I mean, we have to think that, are these adapted the same way in California as they are in Wisconsin to deal with these conditions? It's no wonder that we're seeing the variability in performance, perhaps, and think through, and I think that's interesting in the study that they're doing with transitioning to mature organic farms. How can we foster the native soil microbes to perform the function we want that are probably better adapted to those soils anyway? And what are their role in transitioning land versus mature organic land? And how do we kind of adapt our practices as we move forward with these regenerative soil practices? So it's complex. I know it's frustrating not to have a firm answer, and that's, again, the shift in mindset between conventional and organic, but there isn't a one-size-fits-all answer. And I think these trials that Rick said are so important. I would say, though, not in the corner of the field, because the corner is not represented. You want it on your best ground. So you want to put it in the middle, and you want to collect the data. You just don't want to look at it and say, I think that looks better. You really want to collect the yield and other data, too, forage analyses. I mean, look beyond just the yield. So we have a quick transition to the next talk, but Rick and Teal, you're going to be around for a little bit longer?
I have to run.
You have to run, okay.
Teal will be here in that
Okay, actually, were we supposed to end at 10:30? Oh, we have a couple, I'm going to keep you here for a couple more minutes because I think there might be a couple more questions. Back here first and then you. I want to make sure we take advantage of you for as long as possible.
So is there a test to find out what mycorrhizal or what we have out there and then like how populate it is? Is there a cost-effective test?
I'm not aware of a commercially available one yet, although just at a big potato trade industry thing in Austin 2 weeks ago and saw a company out of California that's trying to do this. The technology is there. It's not terribly expensive. I mean, in our lab, we could probably get that information from one soil sample for dozens of dollars if you really break it down per sample. What we lack is knowing what to do with that information. So I can give you a list of the 1,000 kinds of microbes you have in your soil, but I could not tell you if that's good or bad. Like, are you doing well? Are those the microbes you want? Or are those 1,000 terrible microbes that you want to get rid of all of them? That's where it's really lacking. And so I will make a prediction that in the next 5 to 10 years, there'll be a number of companies offering this test because the technology is readily available. And so keep an eye out for that, but also keep an eye out for what are they promising, right? Right now, we do it for the growers who collaborate in our research projects. We always give them that list back, right? As, here's a thank you for.
And I will say, we are recruiting. So unfortunately, and maybe we can expand this, we're doing this in the southwestern region of Wisconsin. We want to find more fields, though. And as Rick said, we'll give you the data back. So if you're interested in contributing to this data set that is part of Teal's study, if you want to talk to Leah or me and give us your names, we're looking for more fields. And again, we'd love to expand this, but right now we're limited to the driftless area of Wisconsin.
What what I always say when I give the data back to growers is treat this like you would a TV psychic, right? They always say at the bottom, for entertainment purposes only. Because we're not sure what to make of the data yet, but it's kind of fun to look at, right? That's where we are. Hopefully we'll be moving towards a more satisfying answer in the years to come.
Kind of a question related to mycorrhizal fungi. In our own experience with some permaculture, raspberries specifically, in the same field side by side, we note huge population differences when we set them out for laboratory analysis and mycorrhizal populations between one and another 10 feet apart. Is that a genetic trait that some plants may have that are more conducive to it versus others?
Yes, they're definitely across different plant species, so different crops are going to be, some of them are really good hosts for the fungi, and so they're like open doors, welcoming in. Others are much more controlled. They don't let them in. Like I said, the brassicas, the cold crops, don't let them in at all. Even between varieties of a crop, there are genetically based differences in how sort of receptive they are to microbial fungi.
We just did a study with organic carrot, and we inoculated it with 9 different. This is where the diversity comes in. I think it was 9 different species of AMF, and we refined it even further. So not even just the species, but we did the same species, but isolates that were taken from North Carolina versus California. We chose different varieties of carrots, so heirloom varieties, open pollinated varieties that are older varieties, as well as hybrids that were bred under more intensive chemical synthetic fertilizer conditions. They all respond differently. Even the isolates collected from different areas of the country that should be the same species of AMF. Depending on the variety of crop you use, they may respond differently. Depending on the year, they respond differently. So there's just a ton of complexity. And again, I don't want to make it seem like as researchers we're being totally dismissing the observations of organic farmers and how they've seen value. We don't want to do that, but we just want to be sure that we just recognize the complexity of these systems, and we're just at the cusp of understanding how these interactions all play together. So, and definitely with all the experience that farmers help collect, we can better understand that, especially when we do this on-farm research. But it's just, as Rick and Teal are saying, it's just incredibly complex.
And just one last point on that question. It could be the variety. It could also be like there are differences in the soil a couple feet away that may be hard to pick up with a typical test, but that the fungi respond to. And the other thing I'll point out, if you're doing these mycorrhizal tests that tell you like how much do I have, it isn't always the case that more is better, right? And different varieties not only differ in how much fungi will colonize them, but how they respond to every unit of colonization. Some of them, a little bit is all they need, and they're better off having a little bit than having a lot. Others, plants need a lot, and so more is better. It's just hard to know.
All right, I had this question asked of me a lot of times when I'm on farms, and so I took and went to a biology school out in Oregon, and Earthford is the company that put it on. It was a 2-day school similar to this, and it was really good. You got to bring your soil, put it under a microscope, look at the biology that's there, and they've got slides with 100 squares, 10 by 10, and you measure so many grams, put it in water, take a drop, put it on that plate. So it's pretty intense getting that number of how many flagellates, cellates, what the measure of your hyphae is, all those things that are totally unpredictable because you got tillage involved, you got different weather scenarios. So I sent in 2 samples. When I got back home, I couldn't resist. So I sent in 2 samples. They were fairly expensive. And that farm had been organic since 96, so it was 20 years organic, treated very well with cover crops, not a lot of bought-in inputs, mostly cover crops and just minimal applications, so he's yielded well. All my biology was at the top of the graphs, and all I did was put some compost on it and carried on like I've been doing. But I would like to see some kind, if you're going to do this research, why don't we have some kind of number where we need to be? You should find what is low, because there's going to be farms that are low, maybe soil that's a little wetter, maybe sandier soils. Those are places where you put fertilizer on, you can see the difference. You know, a lot of times on good soil, it's hard. You got to put a lot on to see a difference. You know, and just because you don't see it, don't mean it's not there. Look at UC Davies did a pasture study, Joe, and we sponsored that, didn't we, at Organic Valley? And then, all summer long they couldn't see nothing. But when they cut and measured, the difference paid for itself 3-4 times. You know, so the naked eye is not going to tell that. If you're not going to do an honest test plot, you know, do you really want to do it? Because you might just be kidding yourself.
That was my point, Daryl. And really take the time to collect the data, and the data that is meaningful for you. So it may not be just yield, it may be forage quality, but if you're going to bother to do it, as Daryl's saying, take the data. The naked eye isn't enough. Rick, do you have any comments about Daryl's before? I probably should close it at that point. But
I would love to be able to give that number. What we're trying to do is, the gulf we have is between the things you can measure, all those ciliates, the height, whatever, and some functional number. Like what we want that community to do is, well, ultimately grow a big crop profitably, right? You can step back a little bit and say we want a crop that has all its nutrient needs met, a crop that has low disease, or whatever. So what we're trying to do in our research is figure out that relationship. How much of this do you need to get this outcome? And if we get a handle on that, then we can put that number on it and say, you're low, you're high, I would invest here or there. That's why we do the kind of experiments that we're doing, right?
All right, thank you, Rick and Teal, and thank you for all your questions as well.